We're starting chapter 2 today, and chapter 2 is all about the molecules that make up the cell. So I've divided it so that we talk about the small molecules and then those small molecules that make up the larger molecules. So this chapter will sort of be divided. And when we get to talking about the lipids, that'll be with the membrane. When we talk about the amino acids, that'll be with the protein chapter. So it's a little bit mixed, but we're going to get through pretty much the entire chapter, with the exception of those pages that I told you that you all need to, that I think is information you should have already known, and that you'll be tested on during the mock exam for extra credit. So those pages I listed, and review these pages in your textbook to prepare yourself for this lecture, for all of cell biology, and in particular for extra credit in your mock exam. Okay, so we're going to start off talking about the small organic molecules. What does it mean to be an organic molecule? Yeah, carbon-based, carbon backbone, as opposed to, the normal everyday use of the word organic, which has nothing to do with what we're talking about in this class. So nothing to do with pesticides or anything like that. We're just talking about carbon-based molecules. The organic molecules are what make up the cell, essentially. So in the small category, we're talking about a molecular weight of about 100 to 1,000 daltons, which is about no more than 30 carbons as far as the backbone is concerned. So there are two main roles for these small organic molecules. The first is they're an energy source. We don't talk too much about that, but it is true that your cells take up things like saccharides, glucose, for example, as an energy source. So they break open these molecules during metabolism specifically, and they utilize that energy of breaking those covalent bonds to store that energy, usually in the form of ATP. So this is all metabolism. We're not going to go into the details of metabolism in this class. And the cell does the same thing, or similar, for things like nucleotides when it needs to, and amino acids when it needs to. So again, the small molecules are an energy source for the cell. And the second important role that they play is that they are the building blocks for the larger molecules, the macromolecules, if you will. So we're going to get to that in a little bit more detail. So what are the small molecules? The simple sugars, fatty acids, amino acids, nucleotides. I did mention the fatty acids as an energy source, but they are also an important energy source for the cell. But in addition, these small molecules are built up in a stepwise fashion that we'll talk a little bit about, not too much, to make these larger organic molecules of the cell. So the sugars make up the polysaccharides, the fatty acids make up the fats and the membrane lipids. Amino acids, of course, make up proteins, and the nucleotides make up the larger nucleic acids, DNA and RNA. And here's just one example. So take, for example, the macromolecular assembly that is the ribosome, very important for translation. The ribosome is made up of several RNA molecules and several proteins put together. So we start off with the subunits, of course, which are amino acids, nucleotides. Those are added sequentially in covalent bonds to make up the larger macromolecules, in this case RNA and proteins. And then typically via non-covalent bonds, interactions that occur between proteins and the RNA, we get these larger macromolecular assemblies. I usually call them complexes. So the complexes are these larger structures made up of the macromolecules. So this is the example the book gives you. Another example would be say the microtubules I've been talking about in your first, giving you examples of in your first lecture. So the amino acids make up the protein tubulin, and then the tubulin assembles into these larger macromolecular structures or complexes called microtubules. And the ribosomes, microtubules, are large enough that we could see using certain microscopic tools such as the EM and fluorescence microscopy. All right, so here's a picture from your book of an example of the four main families of small organic molecules. So here's a sugar, glucose, an amino acid, a fatty acid, and a nucleotide. So before you guys ask, I will answer. What do we need to know? I'm not going to ask you to draw out specific sugars or amino acids, nucleotide, but I do need you to recognize what they look like. So take this image and you know, figure out which is which. It's right there, but maybe cover these up. And try and see if you can recognize which is the fatty acid, which is the nucleotide, the sugar, amino acid. So know the overall structure of what they look like. Be able to recognize them. Okay, so in a typical cell, your book gives you a bacterial cell, probably E. coli, as an example. And this is true for eukaryotic cells as well, give or take some, depending on the circumstance. But this is overall comparable to most cells. Most cells are made up, by volume, of mostly water. So they're about 70% water. And then the rest, about 30%, is made up of other chemicals that aren't water. Out of that 30%, the majority of those chemicals are organic, with about 1% being inorganic in the form of things like ions. So ions, even though only 1% by weight, play a big role, or I guess I should say, in this case by volume, play a big role in the cell, as we'll talk about later. Okay, so the rest are organic molecules. About 3% of that is the small molecules. 3% of that, 30%, and that's where that 1/10 of the total mass comes from, or the total organic mass. 3% of the 30%. And then the rest, large majority of the chemicals other than water in the cell is made up of these larger macromolecules. Now this is again by volume or even by mass. By number, this would look different, because by number there are a lot more small organic molecules, but because the macromolecules are so big, they make up the majority of the volume or the mass. So is the majority of your cell organic or inorganic material? The majority of the cell. Raise your hand if you think organic. Raise your hand if you think inorganic. Inorganic, right? So the majority of your cell is just pure water, which is inorganic. But if we're looking at the organic stuff, it makes up the majority of, again, that other chemicals besides water. And the same can be true for that. Why is it that ions, even though, as we're going to see, play a big role, they're so itty-bitty compared to the other molecules? There's lots of them, but they're so itty-bitty that by number these would switch, but by volume or mass, these are the breakdowns. Do you need to memorize these numbers? No, you do not. This is an example. And it varies cell to cell. So the breakdown of the macromolecules. The majority of the macromolecules by volume being proteins. Oops. Okay. So, the macromolecules that we'll talk about, these are polymers. What is a polymer? A polymer is a large molecule that grows by the addition of a monomer onto one end of the polymer chain. So, usually the subunits, the monomers, are added in a particular sequence. This is true for DNA, for example, for proteins. And the addition occurs via a chemical reaction, because anytime you covalently link two things, or break that covalent bond, we're talking about a chemical reaction that occurs. And the chemical reaction needed to add one subunit to the growing polymer is called a condensation reaction. So many biological transformations, not just a growing polymer, actually occur via condensation reactions. And what I mean by biological transformation is in biology a molecule becoming something else. So this is true for growing polymers. This is also true when we talk later about things like phosphorylation, which is the addition of a phosphate group, or glycosylation, which is the addition of a sugar molecule to something like a protein or a lipid. This is going to occur via a condensation reaction. Okay, so what's a condensation reaction? It's essentially a removal of one water molecule in the process of forming this covalent bond right here. So we remove an H from the growing polymer and OH from the subunit of water total, and we get this covalent bond. So a polymer, I encourage looking at the word, even though we're going to be looking at these big old words that seem like they don't mean anything. But if you take these words a lot of times and break them down, you can figure out what the word means. So polymer, poly, lots, then monomer, one. So monomer makes up the subunits of a polymer, and what's mer in this case is mer is referring to the subunit. So polymer, many subunits, monomer, one subunit. Dimer, how many subunits? Two. Yeah. Trimer, three, etc. So here's a condensation reaction. This is just looking at A and B to form a single molecule that's covalently linked. AB requires a condensation reaction. This is usually energetically unfavorable, so the making, so bringing together two molecules to make one molecule via a covalent bond is usually energetically unfavorable, requires some form of input of energy. And then in order to break this dimer back into its monomers, the reverse reaction is required. So usually that requires an input of water in a process called hydrolysis. So hydro, water, lysis, anytime you see the root word L-Y-S, you can think of breaking apart something. L-Y-S or L-Y-T. Lit is another one. A lot of times hydrolysis reactions are energetically favorable. So this requires energy, this releases energy. And so that's why we break apart molecules in catabolism or in metabolism in order to conserve that energy obtained from hydrolysis. So things like polypeptides or proteins, oligo and polysaccharides, nucleic acids, in order to make them, it requires energy. We grow a polymer from individual monomers being added. And so here's three examples of that, three pictures of that. So for polysaccharides, shown here, this is glycogen, which is a polysaccharide. This polysaccharide grows by the addition of individual monomers, which in the case of polysaccharides is monosaccharides. Here's the condensation reaction, which is also called a dehydration reaction. Condensation reactions, dehydration reactions mean the same thing in biology. So there's a removal of water as the covalent bond is formed, and it requires energy, so this little yellow box is just reminding you that it requires energy. Usually energy in the form of some sort of nucleoside triphosphate, which if you don't know what that is, we'll talk about it this week. You'll know what that means. Usually in the form of ATP or something similar. Here's the making of a protein. Here's the polymer, and here's the monomer being added. Again, condensation reaction, and energy is required. The making of nucleic acids, this is RNA. Nucleotide being added, water being removed, and now we have a new, longer RNA molecule here on the right. And notice how all of these are occurring by the addition of one end of the polymer. Okay, we're going to first talk about carbohydrates. So the monomers of the carbohydrates are sugars, also known as monosaccharides. So we know that sugar is an energy source. Here's two examples of sugars. Here's glucose, here's ribose. But in addition, they also form the monomers that make up the larger polysaccharides and also the oligosaccharides. In your book, you'll usually see the monosaccharides depicted and the polysaccharides depicted as these ring structures. They can also be depicted, although not as often, as these linear structures here. And in solution, monosaccharides are continuously opening and closing linear and then forming rings, linear and then forming rings. We label the carbons. Here they are labeled 1, 2, 3, 4, 5, 6. So this glucose is a 6-carbon sugar. In the ring structure here, these carbons, they're not shown. They're shown as just the points in the ring. So each point in the ring is a carbon labeled here. And again, in solution in water, carbon 1, which is that first carbon here, in this case it's an aldehyde. Either the aldehyde or the ketone. The ketone's not shown there, but this is true for the aldehyde or ketone. Carbon 1 here, there's a reaction that takes place in water where it'll basically bind to the second to the last carbon, so in this case either carbon 5 or carbon 4 in this case, forming this closed ring structure. So here's carbon 1 with the aldehyde group. So this oxygen is the aldehyde oxygen there. And so what we have is C1, 2, 3, 4, 5, and then 6, or C1, 2, 3, 4, 5 in the case of the 5-carbon ribose. Okay, so isomers. What are sugars, essentially? Does anyone know the chemical formula for sugar? CnH2nON. So basically, carbon H2O is the formula. So that's where the name comes from, carbohydrate. Now, what's kind of tricky about some of these carbohydrates or some of these sugars is that they can be found in isomers. What does that mean? What's an isomer? An isomer is two chemicals, two molecules that have the same chemical formula, CnH2nOn, CH2O, but different structures. So here are 3 examples of 3 isomers: glucose, galactose, mannose. All 3 of these guys are 6 carbon sugars with the chemical formula C6H12O6, but they look different, and we can see here where they're different is highlighted. So here's glucose. Galactose here has this hydroxyl group facing up instead of down as compared here to glucose. Mannose has a hydroxyl group facing down here in carbon 4, but in carbon 2 right here, the hydroxyl group is facing up instead of down. So structurally, they are different, even though they have the same chemical formula. Do you think this makes much of a difference? What do you think? Yes, so it turns out glucose, galactose are dietary sugars. If we ingest glucose and galactose, they will pass through to our bloodstream, and then once in our bloodstream, they will go to cells and basically feed our cells as a chemical energy source. Mannose, however, can't bypass. It's not a dietary sugar. If we ingest it, it'll just be waste. So let's look at the difference again. It's minor, very minor, but our bodies have ways of recognizing these two. They have transport systems that allow these 2 sugars, and only this tiny little difference right here is enough of a difference so that mannose is not recognized in that same way. So structure, function, even tiny little differences in structure can make a big difference. Okay, so let's talk about beta versus alpha. I told you the direction pointing up versus down of things like this hydroxyl group are important. The hydroxyl group is attached to the carbon here. And these have a name. So when the hydroxyl group is pointed up, that's called the beta position. When the hydroxyl group is pointed down, that's the alpha position. In solution, in water, remember the rings are forming and then opening up. They can open up again and then they form again and they open up. This occurs just kind of randomly in solution. And when this occurs, the hydroxyl group on the carbon that carries the aldehyde or ketone can rapidly change. So as it's opening and closing, it can go from the beta to the alpha position randomly as well. However, as we're going to see next, when 2 sugars link up in a covalent bond, the alpha or beta position, whichever one it happens to have at the time of the linking, then that stays locked into place. So here's an example. Here's 2 monosaccharides, glucose and fructose. They're going to come together in a, what type of reaction? Condensation reaction, dehydration reaction. So waters eliminate. This bond is a covalent bond. And now we have a disaccharide. Linking, or I shouldn't say linking, but freezing the alpha or beta position into place. So this is facing down, so that, what, alpha or beta? alpha is now frozen, basically there in the alpha position. Unless we break this bond, and then the monosaccharide is allowed to do the thing where it opens and closes again. But now that this bond is formed, these rings are no longer available to the glucose anymore. So, glucose and fructose make the disaccharide called sucrose. And here we have just 3 common disaccharides. Maltose, glucose plus glucose, lactose, galactose plus glucose, sucrose, glucose plus fructose. Notice all of these have glucose as one of the 2 subunits. And sucrose is shown here. So if we put 2 isomers together, so as an example here, glucose plus glucose makes maltose. Glucose plus galactose makes lactose. All of these are isomers of each other, which makes maltose and lactose have the same chemical formula, right? And therefore maltose and lactose are also isomers of each other. Yeah?
I'm just a little confused because I thought you said when it was a down hydroxyl that it's an alpha.
Mm-hmm.
But why is that beta fructose? I shouldn't see it.
So that's a good question. This right here is named for that's a good question, and I have the answer. Why is it beta? Oh, because of this. So we're looking at this right here. Don't worry too much about that, but it's because here's carbon 1 and this is facing down. I'm only telling you about alpha and beta, and you'll see why in a minute. So it's named after carbon 1, and it's named after which is the higher molecular weight. And in this case, it's this one. Usually, if we're just looking at H versus OH, it's the OH. Does that answer your question? Okay. All right. So let's take a look at a glycosidic bond. So formation of a disaccharide occurs between monosaccharides. We have a condensation reaction that occurs, and the bond between 2 monosaccharides to form a disaccharide is called a glycosidic bond. Now, to break the glycosidic bond requires a hydrolysis reaction, the input of water. So again, root words, saccharide right here, saccharin, sugar. So anytime you see that word, think of sugar. In addition, we have glyc. Glyc. We're also going to be referring to sugars or carbohydrates whenever you see that word glyc. So a glycosidic bond, the bond between 2 sugar subunits. So sugars or carbohydrates are special. They're special when they form these polymers, and they're special when they link up, forming these glycosidic bonds. And the reason for that is because unlike the amino acids, which we'll talk about next, and the nucleotides, which we'll talk about next, the way that they can link up is really diverse. And here's an example here. If we're just looking at 2 identical glucose subunits coming together, forming glycosidic bonds, the way in which they can do this is illustrated right here. We have 11 chemically distinct possibilities for a disaccharide made from 2 glucose units. It's always with carbon 1. So carbon 1 is always included. So which is carbon 1 again? The carbon that's linked to this oxygen right here. So carbon 1 can link up with carbon 1, carbon 2, carbon 3, carbon 4. Not carbon 5. Can anyone tell me why not carbon 5?
Which is 1, 2, 3, 4, 5, which is the one right here.
Doesn't have an O.
What?
Doesn't have an O.
Not because it doesn't have an OH. It doesn't, you're right. Does anyone remember the carbon binding rules? Yes, carbon can only bind, covalently bind to 4 things. It can only, not to 4 things, I mean that's true, but it can only have 4 bonds associated with it, is what I mean. So, carbon 5 is already bound 1, 2, 3, and then the H is not there, but the H is not shown, but it's there. It's already bound to 4 other things. But carbon 1 can bind to 1, 2, 3, 4, and not 5, and 6. In addition, depending on whether it's alpha versus beta, this difference is important. It's going to be important for things like other proteins recognizing it, etc. Depending on whether the glucose is in the alpha or beta position, that carbon 1 is in the alpha or beta position, that's going to create a different covalent bond. So here it is in beta, so we can covalently bind beta 1,6. The 6 is the other carbon. Beta 1,4, beta 1,3, beta 1,2, beta 1,1. And then in the case of 1, that carbon can be alpha or beta. So we can have beta 1, beta 1, or beta 1, alpha 1. Because that second monosaccharide, its carbon 1 is also in alpha or beta position. And then again, if it's an alpha, sticking down here, it can go to 1, 1, 1, 2, 1, 3, 1, 4, 1, 6. So all of these are true, giving it 11 distinct possibilities. What does that mean for when we start getting into larger? So we're going to talk not just about disaccharides, but oligosaccharides and polysaccharides. What does that mean for the molecules when we start getting into these larger molecules made up of subunits that can link in all these different ways? And in addition, we're not just going to do glucose as a subunit. We have all kinds of other sugars that can link up to make up polymers, oligosaccharides, and polysaccharides. So what does it mean for these molecules? Do you think? Anybody? What does it mean that we can do these weird linkages? Not just make it a straight molecule that's always linking to the same thing, like we would for nucleotides.
That can give them different structure.
Different structures, yeah. Diversity of these oligosaccharides and polysaccharides, heck, even disaccharides, diversity is going to increase by a lot. Yeah.
So when, like, on the right side where it says alpha 1 to alpha 1, for the second one, why doesn't it say alpha 1 to alpha 2? Because it's on the bottom.
Because you are denoting only the carbon 1. So when you do this type of thing, you name the linkages. You're naming the alpha and beta versus just carbon 1. So if it's 2, then you're not saying whether it's alpha or beta 2. It's just 2, if that makes any sense. This is the naming designation that's given. You name the 1, whether it's alpha or beta, carbon 1. So that's why these don't have alpha or beta associated with it. It's carbon 2, carbon 3. Okay, but this slide just illustrates how these carbohydrates are special, these polymers are special, in that there's such great diversity found within because of this property. In addition, because we can link to a number of different carbons. So glycosidic bonds can occur in a number of different carbons. We can also get branching. So if we add in a third sugar monomer here, it could link right there, and now we have a branched molecule. We don't have branched molecules, we don't have branched nucleic acids or branched proteins. All right, so we have the regular carbohydrate sugar monomers, monosaccharides, which are basically carbon with water, CH2O. We also can have these sugar derivatives, which are equally as important in biology. And the sugar derivative is essentially the sugar, the monosaccharide, with an extra functional group attached to it. So here are 3 examples. Now again, I'm not going to have you draw these or anything like that. You just have to recognize when a sugar is a sugar derivative. In other words, it has an extra functional group attached. So here we have glucosamine, and again, the names give away what they are. Glucosamine is glucose with an amine group attached to it. Glucuronic acid, shown here, is glucose with a carboxylic acid attached to it. So, replacing that hydroxyl group, the amine group, replacing this right here is the carboxylic acid. Replacing this hydroxyl group in this case is an acetyl group and an amine group. So here we have N-acetylglucosamine. We have glucosamine as here, and then the N-acetyl part comes with this acetyl group linked to the nitrogen of the amine group. So we have N-linked acetyl on glucosamine, basically. Or because this is a larger of the names, we abbreviate this either NAG, N-A-G, or as I like to abbreviate it, GLIC-NAC, because it's funner to say. So GLIC-NAC is written out like this. Okay, so and we'll get back to these. We'll see examples of these in different biological molecules. Okay, so if we have, beyond the disaccharides, if we have monosaccharides making larger polymers, now we're getting into the oligosaccharides and polysaccharides. So oligosaccharides. Oligo means, multiple, so more than 2 would make it an oligosaccharide. Poly means many. So these are designations, the cutoffs are arbitrary, but about 10 or less monosaccharide subunits makes up an oligosaccharide. Polysaccharides usually build these really large molecules. So, and we're talking hundreds of individual subunits or more, depending on how large the polymer is. So really, if the if the polymer had about 11 monosaccharides, I would probably still consider it an oligosaccharide. So the cutoff isn't exact, but it's just the larger ones are the polysaccharides. Shorter chains of monosaccharides are oligosaccharides. So, glycogen is shown here. Glycogen is a polysaccharide made up of glucose subunits coming together. Here's an EM image of these glycogen granules in a liver cell. So glycogen, you make glycogen in your body. Does anyone know what glycogen does? What it's used for?
Stores.
Yeah, basically it just stores glucose in this larger polymer so that you have some glucose available when you need it. So the idea is that if you eat something with less complex, simpler sugars, those are not too easy to break up, not too difficult to break up, pretty easy to break up into the smaller subunits, which your cells can take in and have a ready available energy, pretty easy available chemical energy in the form of things like glucose or sucrose, which is a disaccharide, that the cell can easily take into the cell. Your body can easily take into the cell and break up to make energy. Now, if you run out of that easy available simple sugar energy, monosaccharides, disaccharides, your liver cells store these granules of glycogen, which is this polymer of glucose that the liver can then break up into smaller glucose subunits and release into your bloodstream, therefore bringing your bloodstream back to normal glucose levels. So your liver cells, your muscle cells also need lots of available energy, and so your muscle cells also contain glycogen stored. Just in case your glucose levels in your blood go down, your muscle cells can break these glycogen polymers into individual glucose subunits. Okay, so that's important, right? This glycogen. And the molecule is shown here. So glycogen, these little blue things are the individual glucose units. And you can see here, because of the properties I told you about how monosaccharides can link up, you can see that this polymer is branched. So this glucose right here is covalently linked to 3 other glucose subunits, making this a branched. And so is this one right here, and this one right here. At every branch point, we have a glucose molecule that's covalently linked to 3 other glucose subunits. Okay, so that's glycogen. Over here we have a plant cell. And this plant cell has these structures where it stores up starch. So what's starch? Besides something delicious that maybe you had to eat this morning in the form of potatoes, or maybe yesterday you had some fries with your Super Bowl dinner. What's starch? Yes.
Does it store energy in plants?
It's just energy storage in the form of what? Does anyone know? What is the molecule storage? It is energy storage. And then we eat it and we utilize it as an energy source as well. A nutritional energy source. Starch is glucose subunits linked up together in a large polymer. So it is the plant's version of glycogen, essentially. It is glucose subunits coming together in a large polysaccharide, and it is also a branched glucose polymer. So what's the difference between starch and glycogen? Is the branching. So alpha versus beta, which carbons, 1 versus 4, which versus 6, I don't remember what the branching is exactly. I could look it up, but it doesn't matter because you guys don't need to know it. But different branching patterns, alpha versus beta, etc., gives you 2 different molecules of essentially the same thing. Glucose, branched glucose polymer. But depending on how exactly that's done, gives you 2 different molecules. Now a third molecule, a third polymer, polysaccharide, is also glucose subunits, a branched polysaccharide made up of glucose subunits, branched in a different way, a third type of way, and that gives you a third polymer, which is the most abundant organic chemical on Earth. Does anyone know what the most abundant organic chemical on Earth is? Cellulose. It's found in, it's not a glucose storage polymer, as is glycogen and starch. Instead, it is a plant cell wall material. This is cellulose. It's basically glucose subunits branched into a polymer. And now we have, and why is it the most abundant organic chemical on Earth? Because all the plants that are alive right now, the trees and any of the other plants, plus all of if you guys are wearing cotton, you guys have it on you. Plus if you're writing on paper, which some of you are, that's also, plus we're inside of a building made of wood, somewhat made of wood, and so basically that makes it the most abundant organic. Three different polymers, three slightly different uses, and definitely cellulose is completely different use, right? It's structural versus energy storage. And again, all from glucose subunits, just the way that they link up and branch is different, making them different molecules altogether. Okay, so an example of a sugar that can either be a polysaccharide or an oligosaccharide, depending on how many sugars are involved. So a category of sugars are these glycosaminoglycans. So, glycosaminoglycans, again, the name might kind of jot your memories to what's happening. Glyco, two sugars, amino, an amino group associated there with it. And so what glycosaminoglycans are these repeating disaccharide units, as shown here. So here we have, this is one example, this is hyaluronic acid. And here we have glucuronic acid. I showed you a picture of that earlier. Linked to N-acetylglucosamine, linked to NAG. And this repeats, here it is again, throughout the length of the molecule. So however long this molecule is, it's just a repeating disaccharide unit. Glucuronic acid, NAG, glucuronic acid, NAG, glucuronic acid, NAG, glucuronic acid, NAG, NAG. What's it doing? We'll talk about on Wednesday. Right? Because we're done? Yes. Okay.